So we've done a lot of glomerular anatomy, and it was mostly to understand what are the things that prevent protein from being filtered. But it is complex and hard to remember, so I wanted to go over one thing that might help. You might notice that if you trace along here, along the epithelial cells of Bowman space, you can actually continue along to the podocytes and then get back to these epithelial cells over here. So all these things are continuous with each other. And so that might actually give you a little hint as to how this whole structure was formed. So initially what you had in development was these epithelial cells just forming a blind pouch. And so this is the beginning of the nephron, and it continues down that way. And you had some capillary cells up here with the arterioles, the afferent and the efferent, and all of that held together by your mesangial cells. And so as you can guess, this structure just pushes down into this one. And so after that happens, you get a picture that looks more like this, with these capillaries pressing in here. And let's keep drawing those arterioles and the mesangial cells. And then eventually it pushes in even more until you get something like this structure here. So basically these cells here that are up against this little vascular tuft start to change and become like these podocytes. And so hopefully that helps you sort of understand how this layer is continuous and how these podocytes cover every aspect of this capillary tuft, including the mesangium. So as you can see, the podocytes also line the mesangium that's exposed, not just the capillaries that are exposed. Now there's yet another kind of cell that we haven't drawn on here that's really important. And that's a cell that's going to line this whole space that I'm drawing here. So it's going to go around the outside of this whole clump of capillaries and mesangial cells. But these cells look kind of weird, and that's why I didn't just draw them right away over here. To show you what they look like, let's actually go over to this drawing over here. And so here's the cell body of one of these cells that we're talking about. And what's cool about them is that the way they cover these capillaries is that they have these arms that reach out, and then those arms have these little fingers that wrap around the capillaries and cover the glomerulus. So each arm has a lot of these fingers. And what's really cool about these guys is that there's multiple of these cells covering one glomerulus. So let's draw another one over here. Here's another cell body. And what's really cool is that they may actually reach out and intertwine their fingers. So here's another arm, and I'm showing that the fingers from this arm are intertwining with the fingers from this arm to help cover the whole glomerulus. So let's draw one of these cells over here on this diagram. So here would be its cell body sitting on top of this capillary mesangium clump. And then here would be some little fingers maybe coming off the arm of that cell. And I'm just showing kind of the intersection of those fingers as you see them, because we're cutting through to make a 2D picture here. Then maybe here we have another cell body and some more little fingers visible. So let's finally name these guys. What are they called? Podocytes. So why are they called podocytes? Well, pod means foot, like you can think of podiatrist. And these little things that I was calling fingers are actually not called fingers, even though that's what they kind of seem like to me. All these little things are actually called foot processes. So they're little foot processes that come off of the podocytes.